Welcome back to the deep dive. You handed us a pretty dense chapter here. Chapter
Oh,
2, chemistry from microbiology for the healthcare professional. We get why you need a quick way through this. So our mission today, just reciting definitions and really translate this stuff, protons, bonds, molecules, into understanding how microbes work, infection, how drugs actually function.
exactly. You really can't skip this. Chemistry is, well, it's the language of life down at the cellular level. If you miss this, you won't grasp why an antibiotic hits a specific target, or how pathogens communicate, or even how molecules get across a cell membrane. It dictates structure, function, and critically, how well drugs work.
Yeah, and before we dive right into atoms, it's worth taking a quick look back. The sources mention some fascinating history. For centuries, Western medicine was kind of stuck with folk remedies and this ancient Greek idea of the four humors: blood, phlegm, yellow bile, black bile.
Right, pushed by Hippocrates and Galen, treatment was all about balancing these non-chemical fluids.
Until Paracelsus shows up in the 16th century, he was a real game changer.
Totally revolutionary. He argued the body, disease, it's all fundamentally chemical, so treatments should be chemical too. And this led him straight to that principle that still defines pharmacology. All things are poisons. It's only the dose which makes a thing a poison.
That insight that the effect is tied to the chemistry and the amount, that's what modern healthcare is built on. It connects directly to this key idea we'll keep hitting, the chemical molecular structure, or CMS. You often hear it called the lock and key model.
And it explains how molecules recognize each other, how they bind, how they interact with specific targets. Could be a hormone, an enzyme, or a drug targeting a microbe. Okay, let's really unpack this, starting right down at the atom.
Okay, basics first. Matter. Occupies space, has mass. Made of elements, things you can't break down further by, natural means. Smallest particle, the atom.
And for microbiology, you really need to know the big six: oxygen, carbon, hydrogen, nitrogen, phosphorus, and sulfur. Remember those. OCHNPF.
So inside that tiny atom, what's the structure? You've got the nucleus, right? Dense center.
Yep, holding the positive protons and the neutral neutrons. The proton count gives you the atomic number, that's the element's ID tag. Protons plus neutrons, that's the atomic weight.
And buzzing around the nucleus.
That's where the negative electrons live, in specific energy levels or shells. And this is where all the chemical action happens. Atoms want stability, which usually means filling that outermost shell. Typically 8 electrons, though that first shell only needs 2.
And those electrons in the outermost shell are the key players.
Absolutely, they're the valence electrons. They determine everything about how an atom will bond and react chemically.
Okay, identity, stability. Got it. But here's a little twist the sources mention. Isotopes.
Ah, yes. So you keep the same number of protons, same element, but you vary the neutron count.
Which changes the atomic weight.
Exactly. Like hydrogen, normal hydrogen, no neutrons. Deuterium has 1, tritium has 2. Same element, different weights.
And some of those heavier ones, like tritium, they're unstable.
Right, they're radioisotopes. They decay, releasing energy and matter. That's radioactivity. And while radiation can be dangerous, we actually harness this in medicine. We can tag molecules with radioisotopes.
You can track them.
Precisely. Researchers track synthesis pathways. Clinically, doctors use labeled molecules to find damaged tissues with imaging scans. It's pretty neat nuclear physics helping us see inside the body.
Now, what if an atom loses or gains one of those crucial valence electrons?
Then it gets an electrical charge and becomes an ion. Lose an electron, you're positive a cation. Gain an electron, you're negative an anion.
Cations positive, anions negative.
Got it. And these ions are the basis of electrolytes. These are substances that, when you dissolve them in water, they just break apart into free ions.
Letting the solution conduct electricity.
Exactly, an ionic solution. And the source material really emphasizes this because complex life needs a super precise balance of these electrolytes. It maintains the osmotic gradient, which is critical for, well, everything: nerve function, muscle function, homeostasis.
Makes sense. And clinically, that's why rehydration isn't just water. It's about getting that exact ionic balance back with solutions containing sodium, potassium, chloride. It's applied chemistry keeping people alive.
Okay, so we have the building blocks: atoms, ions. How do they stick together?
Chemical bonds.
Right. Atoms bond to fill that outer valence shell. Remember carbon. It can form up to 4 bonds. That's why it's the backbone of all the big organic molecules. Huge versatility.
And the type of bond is really important for strength and how the molecule behaves, right? Let's maybe rank the main ones, strongest to weakest.
Good idea. Strongest first. Covalent bonds. This is all about sharing electrons. If they share equally, it's nonpolar. Very stable. Think of those long carbon chains in fats or sugars, really robust backbones.
But the sharing isn't always equal.
Nope. If one atom, like oxygen, is stronger, more electronegative, it pulls the electrons closer. Unequal sharing. That creates a polar covalent bond. The molecule ends up with slightly charged ends. Water is the classic example. Still strong, but interactive.
Okay, covalent sharing. What's next?
Second, ionic bonds. Here, it's not sharing, it's a full transfer of electrons. One atom gives, one takes. Creates those fully charged ions, cations and anions held together by pure electrostatic attraction, like sodium chloride, NaCl, table salt.
Strong bond, then.
Strong in a dry crystal, yes. But here's the key thing for biology. Put them in water and they dissociate. They break apart easily. So ionic bonds aren't great for building stable structures inside a watery cell. Covalent is the way to go for that.
Right, makes sense. Third type.
Hydrogen bonds. Now, these aren't true bonds like covalent or ionic. They're weaker attractions, specifically between a slightly positive hydrogen atom that's already in a polar covalent bond and a slightly negative oxygen or nitrogen atom nearby.
Like the glue holding DNA strands together.
Exactly that. And they're responsible for many of water's unique properties, too. Weak individually, but powerful in large numbers.
Okay, and the last one. Weakest.
Weakest, but super important, especially for how drugs work. Van der Waals forces. These are very short-range, kind of fleeting attractions between molecules, but they fine-tune the final fit, that precise molecular arrangement.
The lock and key specificity.
That's it. Often the difference between a drug hitting its target microbe and hitting your own cells comes down to getting these weak Van der Waals interactions just right. It's all about the perfect fit.
Okay, bonds hold things together. What about making and breaking molecules? Reactions.
Right, the pathways of metabolism. The cell is constantly doing two things, building up and breaking down.
Building up.
That's dehydration synthesis, or anabolism. You take small monomers, link them into a bigger polymer, and you do that by removing a water molecule. Dehydration. This process requires energy input. It's endergonic.
And breaking down.
That's hydrolysis, or catabolism. Breaking down polymers into monomers by adding a water molecule. Hydrolysis, water splitting. This process releases energy. It's exergonic. Think about when you digest food, that's catabolism, breaking stuff down to get energy.
So, anabolic builds, needs energy. Catabolic breaks down, releases energy.
You got it. And managing that energy flow often involves redox reactions, oxidation-reduction.
Okay, redox, oxidation is loss.
Oxidation is loss of electrons. The molecule becomes more positive.
Reduction is gain.
Reduction is gain of electrons. The molecule becomes more negative. Remember, oil rig. And these two reactions are always coupled. One thing gets oxidized, another gets reduced. This electron transfer is fundamental for capturing and releasing energy, especially for making ATP.
ATP, the cell's energy currency. We'll come back to that. But first, let's talk about a stage for all this chemistry, water.
Ah, yes, H2O,
Yeah.
the universal solvent thanks to its polarity. Those polar covalent bonds and the resulting hydrogen bonds between water molecules give it amazing properties. High cohesion water molecules stick together.
Which leads to surface tension, right? Like how water striders can walk on water.
Exactly. And because it's polar, it's great at dissolving other polar or charged things. Water is the solvent, the thing doing the dissolving. The stuff being dissolved is the solute. Together, they make a solution.
And things dissolve differently based on their own polarity.
Right. Hydrophilic means water-loving. These are polar molecules like glucose or amino acids or ions. They dissolve easily, get surrounded by water molecules, hydration spheres.
And the opposite.
Hydrophobic, water-repelling. These are nonpolar molecules like fats and oils, lipids. They don't dissolve in water, they clump together.
This leads us to tonicity, which sounds important for cells in different environments.
Absolutely critical. Tonicity compares the solute concentration outside a cell to the inside. Imagine a bacterial cell. If the outside solution is isotonic.
Iso means same. So same solute concentration inside and out.
Perfect. Cell volume stays stable. Happy cell. Now what if the outside is hypertonic?
Hyper means more, so more solute outside, like salty water.
Right. Water will then rush out of the cell, following the solute gradient. The cell loses water, shrinks, shrivels up. We call that crenated, not good for the cell.
And the opposite, hypotonic.
Hypo means less. Less solute outside the cell than inside, so water rushes into the cell.
It swells up.
It swells, and if it doesn't have a strong cell wall, like an animal cell, it can actually burst. Lysis. So understanding tonicity is vital for knowing where microbes can survive and for how we culture them in the lab.
Okay, code water, solutes. What about acidity, pH?
Right. So in water, a tiny fraction of water molecules naturally split into hydrogen ions, H+, and hydroxide ions. An acid is a substance that increases the H+ concentration when dissolved in water. It's a proton donor.
And a
A
base.
base decreases the H+ concentration, often by releasing hydroxide ions, which combine with H plus O's, or by directly accepting H plus CO. It's a proton acceptor.
And the pH scale measures this.
Yep, from 0 to 14. Measures the H plus concentration. Lower pH means more H plus O, more acidic. Higher pH means less H plus M, more alkaline or basic. pH 7 is neutral, equal H plus and OH.
And the scale is logarithmic, right? That seems important.
Crucial point. Each whole number change on the pH scale represents a tenfold change in H plus concentration. So pH 6 is 10 times more acidic than pH 7. pH 5 is 100 times more acidic.
Wow. So biological systems must be incredibly sensitive to pH changes.
Extremely. Which is why cells and organisms like us have buffers. These are chemical systems, like the bicarbonate buffer in our blood, that resist changes in pH. They absorb excess H plus when things get too acidic or release H plus when things get too basic. They maintain stability.
Okay, makes sense. Buffers protect against drastic pH swings, especially during infections where microbial waste products might be acidic.
Precisely. Essential for survival.
All right, we've set the stage. Atoms, bonds, water, pH. Now for the big players, the four major classes of organic macromolecules, the large molecules of life.
Built on that versatile carbon backbone. Carbon's ability to form four stable covalent bonds lets it create huge, complex structures, chains, branched chains, rings, amazing diversity.
First up, carbohydrates, sugars and starches.
Generally have that CHO ratio of about 1.2.1. Think hydrates of carbon, their main jobs, quick energy source and structural components.
The basic unit is a monosaccharide, right? Like glucose, fructose, ribose.
Exactly. Link two together, you get a disaccharide like lactose, milk sugar, or sucrose,
Yep.
table sugar. Link many together. You get polysaccharides,
Which can be for energy storage.
like starch in plants, or glycogen in animals and bacteria. Or they can be structural.
Like cellulose in plant cell walls.
Yep, or chitin in fungi. Very tough stuff.
Okay, carbs are energy and structure. Next, proteins.
Proteins are the real workhorses of the cell, incredibly diverse functions. Their monomers are amino acids. There are 20 common types found in nature.
And each amino acid has the same basic structure, but differs in its R group.
Exactly. That variable R group gives each amino acid its unique properties. And the specific sequence of these amino acids in a chain is dictated by the genetic code in DNA read via mRNA.
Functions are everything from enzymes speeding up reactions to structural components, hormones, receptors on cell surfaces, antibodies. Huge range.
And their function utterly depends on their 3D shape, which has different levels of structure. Primary structure is just the linear sequence of amino acids.
But it folds.
Right. Secondary structure involves local folding. Common patterns are the alpha helix, a coil, or the beta pleated sheet, like a folded fan. These are held together mainly by hydrogen bonds.
And then?
Tertiary structure. This is the overall complex 3D globular shape of a single polypeptide chain. It's stabilized by interactions between those R groups, ionic bonds, disulfide bridges, hydrogen bonds, hydrophobic interactions. This tertiary structure is what creates the specific functional shape, like an enzyme's active site.
And some proteins have one more level.
Some do. Quaternary structure is when multiple polypeptide chains, subunits, come together to form a larger functional protein complex. Hemoglobin is a classic example, 4 subunits working together.
But this intricate structure is fragile. Denaturation.
Big danger. Heat, extreme pH changes, certain salts or chemicals can disrupt those weaker bonds holding the tertiary and quaternary structure together. The protein unfolds, loses its specific shape.
And loses its function.
Completely. A denatured enzyme can't catalyze its reaction. Think about a high fever. It's dangerous partly because it can start denaturing critical proteins in your body.
Right. Okay. Third group. Lipids, fats, oils.
The defining feature of lipids is that they are hydrophobic. They don't mix well with water, but dissolve in organic solvents. And energy-wise, they pack a punch, storing more than twice the energy per gram compared to carbohydrates.
The most common type we think of are triglycerides, right? Fats and oils.
Yep. Made of a glycerol molecule linked to 3 fatty acid chains. We distinguish between saturated fatty acids, all single carbon Carbon bonds, straight chains, tend to be solid at room temp like butter.
And unsaturated fatty acids.
These have one or more double bonds between carbons. The double bonds cause kinks or bends in the chain so they don't pack as tightly, usually liquid at room temp like olive oil.
But for microbiology, maybe the most important lipid is the phospholipid.
Absolutely. The heavy hitter for cell structure. A phospholipid has a glycerol backbone, two fatty acid tails, which are hydrophobic, nonpolar, but the third spot has a phosphate group, which is charged and attached to another polar group. So it has a hydrophilic, polar head, and two hydrophobic, nonpolar tails.
That dual nature is key.
Is everything. In water, phospholipids spontaneously arrange themselves into a phospholipid bilayer heads, facing the water outside and inside the cell, tails tucked away in the middle. This bilayer forms the fundamental structure of all cell membranes. It's the barrier between the cell and the world.
And a major target for antimicrobial drugs, trying to disrupt that membrane.
You bet. There are other lipids too, regulatory ones like steroids, four-fused carbon rings, think cholesterol, sex hormones, cortisol, and prostaglandins, local hormones involved in things like inflammation and pain.
Okay, last group. Nucleic acids, DNA, and RNA.
The information molecules. Their monomers are nucleotides. Each nucleotide has three parts: a 5-carbon sugar, a pentose, a phosphate group, and a nitrogen-containing base.
And the bases are the letters of the genetic code, A, G, C, T, U.
Right. Adenine A and guanine G are larger double-ring structures called purines. Cytosine C, thymine T, and uracil are smaller single-ring pyrimidines.
DNA, deoxyribonucleic acid, uses the sugar deoxyribose. Its job is storing the genetic blueprint. It's usually double-stranded, forming that famous double helix.
And the two strands are held together by hydrogen bonds between complementary bases. Adenine always pairs with thymine, AT, and guanine always pairs with cytosine, GC. That specific pairing is key to copying DNA accurately.
RNA ribonucleic acid uses the sugar ribose. It's usually single-stranded, and its main role is carrying the instructions from DNA to make proteins. In RNA, uracil, MAU, replaces thymine, so the pairing is AU and GC.
And one more critical nucleotide-based molecule we have to mention, ATP, adenosine triphosphate.
The energy currency.
Exactly. It's an adenosine nucleotide with 3 phosphate groups attached. The bonds linking those last 2 phosphates are high-energy bonds. When the cell breaks down food, catabolism, it captures the released energy by making ATP.
And then when the cell needs energy for something like building molecules or moving it, it breaks one of those high-energy bonds in ATP, releasing the energy.
Precisely. Breaking ATP down to ADP, adenosine diphosphate, and a phosphate group, is a highly exergonic reaction that powers almost everything a cell does.
Wow.
It's the fundamental way energy is transferred and used in all known life.
Okay, wow, that was a lot, but let's try to wrap it up. Bring it full circle for everyone listening.
So the big picture. Matter is atoms. Atoms bond to form molecules based on filling electron shells. The type of bond dictates structure and stability.
Chemical reactions, metabolism, are about building up. Anabolism needs energy, and breaking down. Catabolism releases energy, often involving redox reactions for energy transfer.
The whole show happens in a watery environment where polarity, solubility, tonicity, and especially pH are absolutely critical. Buffers keep that pH stable.
And the complex structures of life, carbohydrates, proteins, lipids, nucleic acids, are built from smaller monomers, assembled using these chemical principles, with carbon as the versatile backbone.
And the absolute core connection for you studying microbiology or healthcare is that everything in this field comes back to this chemistry. Infection, immune response, drug action. It's all chemical reactions. It's all molecules interacting based on shape and charge, that lock-and-key principle. Understanding this is understanding the mechanism.
Yeah, really well put. And maybe a final thought to leave you with. We talked about van der Waals forces, those super weak short-range attractions that fine-tune the final fit between, say, a drug and its microbial target.
They provide that ultimate specificity.
So, given how sensitive that fit is, how might scientists in the future use really subtle chemical tweaks, changing just a few atoms on a drug molecule to make antibiotics that are hyperspecific? Drugs that only fit the bacterial enzyme, not ours at all, leading to fewer side effects. That's the goal, right? Rational drug design based on precise chemical understanding. It's definitely something to think about as you go deeper into microbiology and pharmacology.
Absolutely. Well, thank you for joining us for this deep dive into the chemistry foundations from your source material.
Yes, thank you for tuning in. Hopefully this helps solidify those core concepts.
We are the Last Minute Lecture team. Keep learning, keep asking questions, and keep synthesizing this crucial knowledge.